Okay, Anumita, we'll start. Hello?
Hi, sir.
Okay. Good morning, participants. Today, the Department of Environmental Science, Rani Rashmoni Green University, in collaboration with Government General Degree College Singur, organizes a one-day international webinar on emerging trends and future prospects of environmental research. In today's international webinar, at first I welcome Professor Ashutosh Ghosh, Honorable Vice Chancellor Rani Rashmoni University, as today's guest of honor. I would also like to welcome Dr. Aniruddha Mukhopadhyay, Professor and Head, Department of Environmental Science, Calcutta University, to chair the plenary session of today's webinars. I welcome Dr. Avisha Chaudhary from National University of Singapore Medical School, who will enlighten us with her talk. I would also like to welcome Dr. Shubham Dasgupta, Oregon State University, USA, for his talk in this webinar. Welcome all the faculties of Government General Degree College Singur, especially from Department of Botany and Geology, for successfully running the Environmental Science Master's course, even in this period of darkness. Welcome to guest teachers of this course, Dr. Priya Banerjee and Dr. Pallavi Mitro of Rabindra Bharati University, incidentally, who are the co-hosts of today's webinar, and due to their enthusiasm, we can organize this program. Lastly, my heartfelt thanks and a most cordial welcome to the participants of this webinar, including the students of Rani Rashmoni Green University, who are our inspiration. Now, I will request our Honorable Vice Chancellor, Sir, Professor Ashutosh Ghosh, to deliver his inaugural address. Professor Ghosh, sir, please.
Thank you, Dr. Chakraborty. Good morning, everybody. Distinguished speakers of the today's webinar, Dr. Avisha Chaudhary, Duke NUS Medical School, Singapore, Dr. Shubham Dasgupta, Oregon State University, Professor Aniruddha Mukhopadhyay, distinguished scientist in environmental science, Calcutta University, Professor Shantanu Chakraborty, Principal of Singur Government College, and course coordinator for the environmental science of Rani Rashmoni Green University, the participants, the teachers, ladies and gentlemen. I am very happy to inaugurate the webinar on emerging trends and future prospects of environmental research. In fact, this is the first seminar or webinar, whatever you can say, in environmental science organized by Rani Rashmoni Green University in collaboration with Singur Government College. I express my sincere thanks to Dr. Shantanu Chakraborty for taking the initiative and arranging the webinar. In fact, we arranged one webinar in chemistry, and so this is the first webinar in environmental science. Actually, we need this kind of webinar, mini webinars like this. As all of you know, the university has started its journey only last year with MSc courses in environmental science and chemistry. The objective of the university is to educate students of various disciplines with a curriculum that encourages and promotes sustainable development. Therefore, the study and research of environmental science would be the part and parcel of all the subjects which are taught and will be taught in this university. In fact, I just would like to share my experience, which I had, last year during the admission of the students in the university, which I felt is that the students are not that much, you can say, interested, I would say, that much interested to take environmental science as the MSc course compared to the mainstream science courses like, say, chemistry, physics, botany, zoology, like this. Because in chemistry we had say about 150, more than 150 applicants, nearly 200 applicants, but in environmental science we had only say 50 applicants. We have 25 students, and many students still wanted to get admitted in chemistry, but in environmental science the scenario is a bit different. I think in all universities, in Calcutta universities, in Badon universities, and in all other universities in West Bengal, it is not, I'm sorry to say, but it's true. It's my realization, you can say, in the last year, that it is not that popular as the mainstream subjects in science. Yes, there are several reasons for that. I think the main reason is that we don't have the undergraduate course in environmental science in most of the colleges and most of the universities. In mainstream science, we have always the subjects in the undergraduate courses, and so after that they would like to continue that subject in their master degree. So it's obvious, and it should happen. So I think that we have to overcome this situation. We have to start the environmental science in many degree colleges. I think environmental science is a very interesting subject, and it has a very good prospects in future. So we need to start the environmental science in the undergraduate level in many colleges. In fact, I would start, in Rani Rashmoni Green University. It is my plan to start the undergraduate course in environmental science very soon. This is one of the reasons, I think, and other reasons which I felt is that the students are not aware what they will do after passing the master course in the environmental science. Because environmental, in the mainstream science or mainstream arts, the students are interested to get job in school and colleges. They are thinking that the getting job in school and colleges are limited if they study environmental science. It maybe it's somewhat true, but it is not totally true. There are a lot of scope for the students to get absorbed in colleges and schools. But the one thing I think in environmental science, the students will have better opportunity than the mainstream science subject is that in research. There is a lot of scope of research in environmental science. So if you are NET qualified, you can do research anywhere in India. You can also go abroad for doing research. There are a lot of projects also there. So you can join as project assistant in many projects. So if you think a higher study, doing research, going abroad, I think environmental study, this subject environmental study is a very good subject, and it is a very good choice for you who are studying the environmental science in our university and in other universities. So I would like to ask Professor Aniruddha Mukhopadhyay and also Shantanu and also the distinguished speaker, please say something about the scope of the environmental research, what they can do after passing their master degree, because the students, the student needs this kind of information from you, because all of you are doing research, you are in this area you are doing research, and you are teaching in this area for a long time. And we will also, in collaboration with Singur Government College, we will arrange some more seminars also. There are several distinguished speakers in our country and outside India who are interested to talk with you, to talk about their research. And we'll arrange frequently this kind of seminar for the students of the environmental science, and also the other the students of chemistry are also welcome, because you should also know about the scope of the environmental science, because you can also do research in the field of environmental science, because chemistry is a major part, you can say a major part of environmental science. So a chemist, especially if you have some knowledge in the analytical chemistry and some inorganic chemistry and organic chemistry and physical chemistry also, you can contribute a lot in the environmental science. That's all I would like. That's all I wanted to say. That's all I wanted to say for today's seminar. I wish all the success for this seminar. Thank you very much.
Thank you, Professor Ghosh, Honorable Vice Chancellor, Rani Rashmoni University, for your illuminating talk. Now I will request Dr. Aniruddha Mukhopadhyay, Professor and Head Department of Environmental Science, Calcutta University, to chair the session of today. Professor Mukhopadhyay, please. Hello. Am I audible?
Yes, sir.
I would request Professor Aniruddha Mukhopadhyay to start the proceedings. Aniruddha da.
Yes, am I audible?
Yes, please start.
Yes. Respected Vice Chancellor, Sir, Professor Ashutosh Ghosh, Dr. Shantanu Chakraborty, Principal, Singur Government College, all the faculty members, invited speakers for today's seminar, all the participants, well-wishers, and people who are serving environment. This is a very special occasion when we are organizing this particular webinar at this juncture, because we are passing through a very different kind of phase of our civilization. Now the question is, why environment? Why environmental research? And what is the future prospect of the subject? The question is, environment has become a buzzword nowadays. Each and everyone is talking about environment. The print media, the electronic media, they are talking about environment. When the head of the states of different governments, they are meeting with each other, they are talking about the future of the globe, they are talking about climate change, they are talking about the issues like food security, energy security, all these issues they are discussing. And we are facing some problems connected to environment. In the last year only, we faced many problems. In the last year, in 2020, we faced COVID-related issues. Then you see the locusts, the Locusta migratoria, that species, they migrated from the Middle East, through the Pakistan, they migrated towards India, and they spoiled a lot of, food materials for us. Then we experienced a super cyclone. In the last part of 2019, we experienced Bulbul. In the last year only, we experienced Amphan. And you still remember the devastation caused by these, natural disasters. We experienced earthquakes. We experienced forest fires. And people are talking that, environment has become so important for us. Now we are talking about the supply of oxygen. But we have degraded our forests. We have killed our wildlife. We are facing problems related to water. We are producing huge amount of waste materials. Different kinds of waste materials like, the solid waste, liquid waste, biomedical waste, different kinds of hazardous wastes, plastic wastes, etc., etc. We are going to face huge problem connected to energy, because the store of our coal or natural gas will be finished within a span of, say, 80 years. And we are talking about the climate. We are adding 1.5 ppm of carbon dioxide every year. The sea level is rising. And the temperature is also rising. For the last 100 years, the temperature has risen by 0.64 to 0.74 degrees centigrade. So these are the problems we are facing right now. And for the last 100 years or so, particularly the Western world, they were thinking that something has to be done. They were organizing, seminars, symposia. They were discussing amongst themselves. And the people of the southern globe, they were also organizing different kinds of, movements and other things. But the target of both the dialogue was to keep our environment as it is. I'd like to share some experience. Just a reminder. You see, in 1945, we experienced Hiroshima and Nagasaki. In 1962, Rachel Carson, the great American writer, activist, she wrote a book, Silent Spring. And in that particular book, she mentioned that, well, the use of different kinds of pesticides can kill the birds, and the bird population will be depleted very soon. Spring will be there, but that spring will be silent. In the year 1972, there was a conference at Stockholm, and that particular conference was attended by our then Prime Minister, Mrs. Gandhi. She went there and she spoke about 3 words, and all the words are starting with capital P. P for population, P for pollution, and P for poverty. And this is popularly known as 3 Ps of environment. And in the year 2021, I am adding another word, the 4th pillar of this P, that is P for politics. And that is why when the head of the states, they're meeting with each other, they're talking about the future of the globe. Well, when Mrs. Gandhi came back from Stockholm, after that, most of the environmental acts were enacted in our country. In the year 1972, we had Wildlife Act, Wildlife Protection Act. In the year 1974, we had Water Act. In the year 1981, we had Air Act. In the year 1986, we had Environment Protection Act, popularly known as EPA, or Umbrella Act of Environment. And after that, many rules, regulations. And finally, in the year 2002, we had Biodiversity Act. So we have many acts, we have many regulations. We have different kinds of regulatory bodies like Pollution Control Board. And the Pollution Control Board was established on the basis of the Water Act enacted in the year 1974. And in the year 1992, there was a summit, and this summit is extremely important for me as far as the environment is concerned. And that summit was held at Rio de Janeiro in the year 1992. And 3 things came from that particular summit. FCCC, Framework Convention for Climate Change. Number 2 is CBD, Convention for Biological Diversity. And number 3 is Agenda 21. We were thinking, what will be our agenda in the 21st century, so far as the environmental protection is concerned. And another question was very important, how to protect the biological diversity of the globe and how to protect the globe, because We know about the consequences of the global warming. We know the consequences of ozone depletion, acid rain, greenhouse gases, and all these things are already known to us. And every year we are organizing the COP summits, Conference of Parties. And in that particular summit, the different negotiating bodies, including the governments, NGOs, other stakeholders, they share their views. And that's how we have formed different groups like BRICS, like countries of small islands, etc., etc. And we are actually debating environment and climate. And environment is becoming, very important. Now, what is the scenario of environmental education in India? Before going into that, I want to share just one information that India as a country, as a space, it is just 2.4% of the globe. It is just 2.4% of the globe. And in this particular country, India, our country, we have 16.5% of the global population. So the population is very high in our country, right? And we are getting the energy. Where from? We are getting the energy by burning the fossil fuels. And whatever coal we have in our country, it is just 6% of the global coal. And so far as the fuel and natural gas is concerned, we have just 0.6% in our country. But we are very rich. We are very rich in one thing: biodiversity. 8.1% of the global biodiversity is present in our country. 8.1%. And it is serving many services. It is giving us energy. It is giving us food. It is giving us shelter. It is giving us clothing. It is giving us fuel. It is giving us oxygen. It is absorbing carbon dioxide. So biodiversity has many, many functions. We have to conserve them. And we have a Biodiversity Act in our country, right? Another point is very important. That is, the philosophy of people who are living in the so-called quote-unquote first world, and the philosophy of the people who are living in the so-called quote-unquote third world, there is a difference in that. The first world people, they're telling that no environment without nature. And as because we have more and more people in our country, and primarily those people are living on natural resources, and most of the people are known as ecosystem people. That is why our philosophy is no environment without social justice. So social justice should be there. Environment has many components. It is not only chemistry, it is not only biology, it is not only physics, it is not only mathematics, it is not only economics. Rather, it is the aggregation of many subjects. It is a multidisciplinary subjects. It is an interdisciplinary subject. You have to understand that environment is a different subject altogether. And this very concept is very important to understand what is environment and why environment. So, environmental science has been initiated at the undergraduate level, and this particular introduction, I would say that this is a historical event in our country, because this particular move was initiated by the code. And after that, the same course was initiated at the school level. And now for the last 25 or 30 years, the course has been initiated at the postgraduate level in our country. In different universities, it is being given as a postgraduate degree. As far as Calcutta University is concerned, we initiated this particular program in 2000. After that, the course has been initiated in different colleges. And right now, many colleges are giving environmental science honors course. And hundreds of people, hundreds of students, they join in those courses. And teachers were recruited through the College Service Commission. And many of our students, they are actually teaching in those colleges as permanent teacher. In West Bengal, there are different universities, like Calcutta University, like Kollam University, like Bardhaman University, like Vidyasagar University. We had our course, like, Rabindranath University. But last year only, Rani Rashmoni Green University has initiated this important course. Actually, I was involved since the beginning of framing the course, and the Vice Chancellor said, I must say, he has taken all the pains during the COVID time to initiate this particular course. I must congratulate him that he has taken the initiative. And he is the person when framing this particular course, and he has guided us. All the time he has guided and he has encouraged the entire team so that the syllabus can be completed. And I would like to mention the name of my friend, my younger brother, Dr. Shantanu Chakraborty. He has taken all the initiatives for successfully running this particular course. This is an excellent course, and I think the students will be benefited from this particular course. Anyway, the human civilization, it has got, three parts. The agricultural civilization is about, 10 to 12,000 years old. The age of industrial civilization is about 280 to 300 years, not more than that. And we are passing through a very different kind of globalized information technology-based civilization. The third phase of civilization is very important. And we do not know how this particular phase of civilization is impacting our environment and our life. Maybe the future workers, they'll be able to say something on that. And another point is important. A great scientific writer, Richard Dawkins, he mentioned one particular word, that is mimetics. Meme is the unit of cultural transmission. Because as a human being, we are gathering so many information through technology and other things. This is impacting our environment in a very different kind of way. And that is something which is being regarded as a factor responsible for environmental degradation. I was going through a particular research paper where a great meteorologist, Jeff Thompson, American meteorologist, he has commented that the last few years, the Bay of Bengal only, it has experienced 26 out of the 35 deadliest super cyclones. So every pattern is being changed. The pattern is being changed. There is no doubt about that. Environmental science, we are giving this particular, master's degree as environmental science. What science is very important. Once Einstein was asked to define science, and he told that, well, science is an attempt to make the chaotic diversity of our sense experience correspond to a logically uniform system of thought. That is, by your sense and by your experience, you have to explain the chaotic diversity around you. And that is the basic role of a particular scientist, right? And we are teaching environmental science as a course, where you are learning certain theoretical things, where you are learning certain practical, tools. Now there are different other subjects connected to environment, like environmental technology, environmental biotechnology, environmental management, environmental studies, and different universities, they are giving, these degrees on different subjects. Like in Vishwanath University, they are giving MSc in environmental studies. In Rabindranath University, they are giving environmental studies. In Jadavpur University, they are giving MTech in environmental biotechnology. In IIS WBM, they are giving management degree in environmental management. So there are different kinds of degrees. Anyway, after passing this particular, environmental science MSc degree from your university, you can join, or one can join from any university if he has completed his postgraduate degree in environmental science. He can join the IITs, and he can complete his, MTech degree from there. He can complete his PhD from different institutes, like ISI, Indian Statistical Institute, Indian Institute of Science, ISAR, and different IITs also. Our students from Calcutta University, I know, they have completed their PhD from ISI, completed their PhD from IISc, completed their PhD from IITs, and after passing your, this environmental science course, you can appear for NET examination. NET examination. The UGC is conducting one particular NET examination every year, and you can qualify your NET conducted by UGC. And as because you are also coming from different disciplines like zoology, microbiology, biotechnology, chemistry, or something like that, you can also appear NET examination conducted by CSIR, and you can appear in life science or art science or physical science or chemical science like this. You can appear in the GATE examination. After qualifying in the GATE, you can join the different kinds of PhD programs, or in different kinds of these projects in different universities and institutes. Or you can go for the GRE examination. After giving the GRE, you can go abroad, you can go to USA, you can complete your PhD, you can join in another university as a postdoc, and after that you can join as a faculty member. And now the problem is this, I've talked about this PG courses. The PG courses are this MSc, environmental science PG course. There are different other universities in India, like, Pondicherry University, Pune University, BHU, JNU, Delhi University, Doon University, Tezpur University, Nehu Shillong, then Assam University, Guwahati University, Ram Manohar Lohia University. All these universities, they are giving the postgraduate course. And there are some other related subjects to environmental science. And in those related subjects, this MTech degree is being given by TERI. Delhi, there is a school of advanced studies. They are giving in energy. In Dehradun, there is one institute that is known as FRI, Forest Research Institute. Suppose in Dhanbad, there is one IIT Dhanbad. They are also giving MTech course. And you can do your research in different kinds of fields. Like, this will be your specialization. Like, biodiversity. There is a biodiversity board in our state. They are taking environmental science students as project fellow. The Zoological Survey of India, the Botanical Survey of India, they are also taking fellows from environmental science. So biodiversity and ecology can be your specialization. Then remote sensing in GIS, very important and upcoming subject. And this is a very important focus area of environmental science nowadays. Then waste management. Waste management is a very important area that is coming up in a very big way. And as because I am associated with State Environmental Appraisal Committee right now, I know so many people are now applying for installing the, these biomedical waste facilities in West Bengal or in India. Because after COVID, it has become a very important problem for the disposal of these COVID-related biomedical wastes. Then different kinds of projects related to pollution control, pollution of water, pollution of air, pollution of noise, pollution of other things. So pollution is a very big area of environmental science. Then some kind of remediation kind of research. Remediation, the microbial remediation, the phytoremediation, the chemical remediation. Then toxicology, toxicological researches. That is also a focus area of environment. And another important area that is very, very important, and I would say this is a core area of environment, that is EIA, Environmental Impact Assessment, Environment Management Plan. And for all the industries, the government has made it mandatory that there will be a CSR unit. I mean, CSR means what? Corporate Social Responsibility. And nowadays, another acronym has come that is known as CER, Corporate Environmental Responsibility. So, those environmental consultancies, they recruit environmental science people for doing this kind of EIA or EMP. So, this is a focus area where the students are supposed to know the methods of EIA, the methods of EMP, the different kinds of rules and regulations, the environmental law, all these things are very important for environmental science students. And of course, you can do your research in different kinds of fields. And you can also get jobs after doing environmental science. Right? You can join the environmental consultancies. There are different kinds of consultancies. In West Bengal itself, I know many consultancies who are taking environmental science people as their personnel. And in different areas, particularly in the northern India, they recruit people for environmental consultancies. In different kinds of pollution control board, laboratory, they recruit environmental science people. They recruit environmental science for distilleries, waste management facilities, and different kinds of, this industry. They recruit environmental safety officers. Nowadays, I mentioned that environment has become a buzzword. The print media and electronic media, they're giving reports on environment. And every day, in every newspaper, you will get information about environment. And they recruit environmental journalists with environmental science background. And you can contribute in those newspapers and different kinds of magazines. They recruit people in fertilizer industry. mines and refinery industry, in different kinds of water resource and agriculture-based industry, in pesticide industry, in packaging industry, and also in social planning and urban planning. Why I'm mentioning this urban planning and social planning? Because the government agencies now give huge money to the panchayats and the local urban bodies. That is, these municipalities and corporations. And they do different kinds of activities. And all those activities are connected to environment, like waste management, like water management, like urban planning and all these things. And in India now, 50% of Indian people, they are living in the urban centers. People are coming from the rural areas towards urban centers for their livelihood. So urban planning is a very, very important area. And most of the cities, they are trying to establish different satellite centers. Like in Calcutta, we have Salt Lake or Newtown. Like every city, like in Delhi, they have Gurgaon, Ghaziabad, Faridabad, Noida, all these centers have been formed. So urban planning is very important. And in those urban planning, two things are very important. One is consumption index, and the second one is assimilation index. How much they consume and how much they assimilate. That is the carrying capacity analysis. How much waste they are generating, how much pollutants they are generating. So all these things are very important as far as the life of a particular urban city is concerned. So there are many areas related to environment. And whatever you do, you serve your surrounding, you serve your environment, and you serve your future generation, and you serve the civilization. So that is the beauty of environment. Because all the, scientific workers, people who are living in other areas of knowledge, they are trying to come towards environment. Because environment is an interdisciplinary subject, it is a multidisciplinary subject where everyone can contribute. So it is a duty of all of us who are learning environment. We are all the students of environment, we are learning it. And we can contribute a lot. Not for us, not for our livelihood only. For the sake of civilization, for the sake of humanity, we can contribute a lot. And that is the call of the hour. In environment, all the issues are emotional. All the issues are emotional. All the solutions are technical. But anyway, some of the decisions are coming from people who are working at the administrative level or at the political level or something like that. But anyway, we can do a lot. We can do a lot. So that is a duty. This is number one. Once T.S. Eliot, he wrote few lines. He wrote that, what is the knowledge we have lost in information? We are gathering different kinds of information from environment. What is the knowledge we have lost in information? We have to be knowledgeable. What is the wisdom we have lost in knowledge? Well, we can be knowledgeable, but we have to have some kind of wisdom. What is the life we have lost in living? We are living, but what is that life? So we have to understand the meaning of life. We have to understand the beauty of life. We have to understand the beauty of our surrounding, the beauty of wildlife, beauty of forest, beauty of living, the joy of living. And for that only, we have to contribute in the field of environmental science. And I think, as far as your career is concerned, as far as your livelihood is concerned, it is not very difficult. So, once again, I want to give my thanks to the organizers that they have given me the opportunity to speak something before you. And the purpose of having a green university is immense. And we have to understand why the name green has been given to this particular university. Vice Chancellor Sir has mentioned about the sustainable development. In the year 2015, the UNEP, United Nations Environmental Program, they have spoken about the sustainable development goals. And the sustainable development goal, the target of the sustainable development goals will be made by 2030. So, with that, I conclude my speech, and at the end, I'll just mention two lines. সব সবুজ কি ছড়িয়ে থাকে শুধুই বনে বনে, খানিক বনের মধ্যে ছড়ায়, খানিক ছড়ায় মনে। সুতরাং we have to understand the meaning of this green, the meaning of this word environment, and we have to work together for the betterment of mankind. Thank you very much.
Thank you, Professor Mukhopadhyay, for setting the platform of today's webinar. Your lectures, as always, illuminate us and make us enthusiastic in environmental science a lot. Thank you very much. Now, I will request Dr. Pallavi Mitro, Assistant Professor of Environmental Science, Rabindra Bharati University, to introduce the first speaker of today's webinar. Dr. Mitro, please.
Thank you, sir. Though it's very difficult to come in after such an overwhelming talk from Professor Mukhopadhyay. Dear students, as we have heard from Professor Mukhopadhyay that environmental science is a multidisciplinary subject meant to solve almost all the problems mankind faces today. So it is extremely important to be a smart learner for your future career prospects. Now I would like to welcome Dr. Shubham Dasgupta. Dr. Dasgupta is presently working as postdoctoral fellow in Dr. Robin Tenguin's lab at Oregon State University, USA. He is working on the developmental zebrafish as a model to understand phenotypic and molecular effects of environmental stresses through multiomics approaches. Dr. Dasgupta has completed his MSc in Environmental Science from Department of Environmental Science, University of Calcutta, and he pursued his PhD in Marine Science from Stony Brook University. And he also has worked as postdoctoral fellow at the University of California during 2017 to 2019. Dr. Dasgupta, welcome, and over to you.
Thank you so much for that introduction. Can I share my screen?
Yes, please.
Hopefully you all can see my screen. Thanks for that introduction again. My name is Shubham Dasgupta. I am currently a postdoctoral scholar in the Department of Environmental and Molecular Toxicology at Oregon State University. And it is a real pleasure to present here today, and also a real honor, because as Dr. Mitro said, I got my master's from the Environmental Science Department at University of Calcutta, Baliganj Science College, where I studied under Dr. Rani Mukhopadhyay, and also Dr. Priya Banerjee and Dr. Avisha Chaudhary, who is going to present later. They were my classmates. So it's kind of sort of a reunion as well as a honorable presentation for me. So today I'm going to talk about zebrafish as a model organism for environmental toxicology and human health research. But before that, I'd like to talk to you a little about my journey and how I came to be right where I'm right now. So I got my bachelor's and master's from Presidency College in Kolkata in human physiology, and then my master's from the Department of Environmental Science at Baliganj Science College, as I mentioned. In 2011, I moved to New York to pursue a PhD in marine science with a focus on aquatic toxicology at Stony Brook University there, working under Dr. Anne McElroy. Then in 2017, I moved to a postdoctoral position at the University of California, Riverside, working with Dr. David Voles. This is my lab group here. And here I was introduced to the world of zebrafish. And then in 2019, I moved to my second current postdoc at Oregon State University within Dr. Robin Tanguay's lab to work to further my training in zebrafish, because this is a premier facility for zebrafish toxicology, not only in USA, but all around the world. So why are we talking about zebrafish? A little bit of history about zebrafish. This is a fish that is native to the Indian subcontinent. Particularly, this fish is present, their native land is all around India, particularly in the Himalayas and southern Bengal and parts of Kerala, parts of southern India. This fish is mostly found in the paddy fields and marshes within Bengal. Particularly, as I said, they are found in streams and paddy fields. And the scientific name of zebrafish is Danio rerio. The word Danio is derived from the word dhani, jar utpotti holo Bangla word dhani, যেটার মানে হলো ধান চাষের field থেকে derived. So this is why this is also of importance to me as a model because it is derived from my homeland. So this fish was initially identified in the early 1800s by Dr. Francis Hamilton, and he was describing and finding a lot of fish within Bengal and the Indian subcontinent, and he found this fish in the Kosi River, and he described this fish as the fish with stripes alternating on each side. So why did it become such an important model for developmental toxicology? So this model zebrafish is used as a model for biomedical research all across the world right now. And there are numerous different applications for this fish model, which include regenerative biology. And this is because zebrafish regenerate many of their tissues, including their cardiac tissues as well as their tail fins. It is used as a very important model for cancer research. It's very used as a model for developmental biology to understand how chemicals affect development. For toxicology, for drug discovery purposes, and this is kind of what I'm going to talk about today. It is used as a model to discover the potency of drugs. For immunology, it's a very important model for neurobiology because there are a lot of well-established zebrafish-based assays within that exist in the literature, and we leverage these assays to study the neurological symptoms and a lot of other different applications. But one of the most important is it is used as a very potent model for genetics because you can do gene editing within this fish easily. So using gene editing technologies like CRISPR-Cas and other technologies, you can do gene editing very easily. The ultimate reason why zebrafish is used as a model, used for all this application to study human health effects, is because although zebrafish is a fish, it's still strikingly similar to humans than you might think. So between humans and zebrafish, remember, these all are vertebrates, and 70% of the genes, functional genes within the body, are conserved. So they have a 70% similarity in their genome, where the same genes have the same functions in both humans and zebrafish. And more than 80% of these genes are associated with human disease. So zebrafish is extensively used as a model to study human diseases. And most of these organs are common between the two models, human and zebrafish, except it's difficult to study lung effects because zebrafish don't have lungs, they have gills. But all the other organs pretty much operate in the same pathway or in the same strategy. And this is why it's a very attractive model to study human health effects. There's also a lot of other reasons. Their development is external and rapid. So if you know, human development occurs in utero, within the uterus, but zebrafish development, since they lay eggs, the whole developmental trajectory, they occur outside of their mother's body. So right after fertilization of the embryo, you can monitor whatever the embryo is going through in real time. They have a very short reproductive cycle, so they reach reproductive maturity within 2 or 3 months. These are really small fish, so there is the ease of maintaining large number of fish in the small lab space compared to rodent models, which need extensive facilities. The zebrafish genome is fully sequenced. So similar to the human genome, we know most of what's there in the genome and most of the functions of the gene. And as I mentioned, over 70% of the genes are similar to humans. The embryos are transparent. So this allows non-invasive in vivo imaging, which means that you can image whatever's happening with the embryo without destroying the embryo. And this is very important for the perspective of developmental biology, because you cannot image human embryos when they're developing inside their mother. As I mentioned, these fish are amenable to genetic manipulation. So you can knock in genes, knock out genes, and control the expression of genes very easily. The adult zebrafish are able to regenerate different organs. One of the most important applications is that these fish are amenable to high throughput drug screening. So due to their high number of offspring, so a pair of zebrafish per day can lay up to around 1,000 eggs. So each individual egg can be tested with different drugs, and I'll talk about it a little in a little while. So these are amenable to this high throughput drug screening. This is one example of a genetically modified zebrafish. What you see here is the head of the fish with the different neurons labeled in different colors. So these are labeled with different fluoropores. And what you can do, the application of this is that if you have a neurological disease or if you have a chemical that affects these neurons, you can study the disruption of the neuron by just looking at this kind of fish under a fluorescent microscope, which you cannot easily do in a human or a rodent model. So my area of research is toxicology, and a large array of toxicants have been tested using zebrafish as a model, starting from metals to pesticides to even stressors like radiation, PFAS, flame retardants, drugs, endocrine disruptors, nanomaterials, name it, and zebrafish has been used to test these toxicants. And within my career, I have worked with many of these toxicants. Currently, I'm working with this flame retardants and these PAHs, which are hydrocarbons, but I have worked with many of these other toxicants. What are the different applications that you can do with zebrafish? As I mentioned, you can do chemical screening, so you can screen a large number of chemicals. You can do gene expression studies because, again, from the perspective of human health, the gene expression between humans and fish are conserved. You can do metabolomic studies. You can study the metabolites. You can do gene editing. You can do real-time imaging. You can do enzymology studies. You can do behavioral studies. So as you can, the main summary of this is that you have a lot of application with a fish that is not so expensive to maintain and not so expensive to study. And you can test thousands of different chemicals within a very short period of time. So again, I use many of the strategies such as gene expression, gene editing, enzymology, behavioral studies, dissections with Zebrafish. So I'm mostly largely a developmental toxicologist. So I study how chemical exposures occur, the developmental trajectory, the developmental path. Again, with zebrafish, it's easy to study this developmental path because their development occurs outside of their mother's body. So right after fertilization, when the embryos only have one stage, you can look at the embryos in real time. What you'll see here is the video of the development of the zebrafish over the first 24 hours. And what you will see is that the embryo develops from a 1 or a 2 cell stage to a totally developed embryo with a proper head, eye, and a tail within a period of 24 hours. So the development is really rapid, and therefore you can do this rapid high throughput studies in a very short period of time. In this case, HPF means hours post-fertilization, or hours after the embryos are laid. So as you can see, this is the video of an embryo undergoing development. And this happens, most of these processes are conserved across most vertebrates. So as I said, zebrafish, the early life stage, within the first 24 hours, it goes from the 1-cell zygote to a well-developed embryo. And then it takes around 48 hours to 5 days for organogenesis occurs, so for the different organs to start functioning, and then there's a lot of applications for this adult fish as well. But for chemical screening purposes and genetic studies, which is my research area, we use this what we call this developmental phenotype. So this is a zebrafish. The top figure is the zebrafish at 5 days after fertilization. So this is a fish larvae. But when you treat it with a chemical which is very toxic, you get what this called this phenotype, where you get this bloated embryos, which this phenotype is called an edema. You get some jaw defect, you get sometimes eye defects, you get this distorted axis of the trunk, which is an indication that the embryo, that this chemical is toxic, and it identifies that toxic chemical. So, as I mentioned, our lab has a huge zebrafish facility. What you see here is that we harvest these embryos from the fish, or when they are laid, from this tank. So each of these blue tanks contain hundreds and thousands of zebrafish. And around 10 minutes after they lay the eggs, we collect it using a strainer. And then at 6 hours after fertilization, we use these plates, this 96 well plates, to do chemical exposure. So each well of this plate will contain an embryo with chemicals in them. And within our lab, we have developed this robot to transfer individual embryo to this individual wells of a plate. So as you can see, the video shows a robot transferring an embryo to this plate, and each of the plates will contain an embryo per well. Then we use this technology called bioprinting. So this is similar to your printer, your inkjet printer that we use in offices or homes, except that instead of ink, it basically prints the chemicals. So now you end up with each well of this plate with a different individual chemical, each well of this plate with a different individual chemical, and we can do exposures with almost 40 to 50 plates with hundreds of chemicals and thousands and thousands of embryos. And then after 1 day, we do this assay called a photomotor response assay. So this is a behavioral response to a light stimulus. And then at 5 days, we do a locomotor behavior assay, which is again a behavioral swimming assay to a light response. And these assays, I won't go into details about these assays, but these assays are indicative of toxicity. So this is this assay, if you have a positive hit in a behavioral assay, this indicates that there might be some neurological or neurotoxic mechanisms involved. This is the kind of data we get out of these screening studies. I don't expect anyone to read the axis, but just to summarize, the y-axis contains 123 different chemicals that were screened here, and the x-axis contains these endpoints, which include some of the morphological endpoints like bent axis or defective jaw or mortality, or the behavioral endpoints, which include this photomotor response assays. And if you have a colored cell here, it means that this chemical was a hit from this assay. And based on this, we decide whether a chemical is toxic or not. So I'll now pivot to some of the research that I have performed over the last couple of years. I have extensively worked with per and polyfluoroalkyl substances, or PFASs. These are chemicals that are present within many of our everyday use products, such as cookwares or shampoo or paints or food packaging, and eventually they either end up in the environment from which they ultimately end up in humans, or humans are exposed to them, or they are included in the consumer products such as food packaging, and humans can directly ingest them. So humans are exposed to them, and then it's transferred to the infants through breast milk or cord blood, leading to a possibility of developmental toxicity. And these PFASs are of primary concern all around the world due to the resistance to degradation. So these chemicals are not easily degradable within the environment, which is why they're called forever chemicals. So within my study, when I was at UC Riverside, I did a study with a series of these PFASs. So what you see here, again, I don't expect you to read all of it, but what you see here is that on the y-axis, you see 38 different PFASs here. And on the x-axis, you see percent survival at different exposure periods and percent normal. So whether the embryos were dead or not, or whether the embryos were normal or not. And by normal, I mean that at 24 hours, you see the embryo with a well-developed head and a tail. But if they were not developed well, or there was a delay in development, you see that the head and tail are not developed. So when we screened these chemicals, we saw that one particular PFAS called PFOSA, or perfluorooctane sulfonamide, stood out as being a hit for a chemical hit within our screen. So these, by 24 hours, none of these embryos were normal. So that led us down to the path on what affects this PFAS toxicity, or what are the mechanisms of PFAS effect. So we did transcriptomic studies, which means we did a full gene expression profile using mRNA sequencing. And not to go too much into details, but what you see here in this figures, the blue dots are what we call the differential expressed genes compared to the control embryo. So we have a control embryo, which is an embryo treated with the solvent DMSO. And these are either upregulated genes or overexpressed genes or inhibited genes. So what we saw here was that the PFOSA, when we exposed the embryos to PFOSA, it led to dysregulation of a number of a lot of different genes. And then when you do something called a gene ontology, which takes all the genes and predicts what biological pathways these genes affect, we saw that liver development was impacted. So it led to these PFOSA exposures led to hepatotoxicity. So we measured whether it's kind of difficult to see here, but if you follow the blue arrow or the blue line, we measured the liver area due to after the PFAS exposure, and we saw indeed that when the fish was exposed to PFAS this particular PFAS, the liver development was inhibited, and you see a smaller liver here, which shows that these kinds of chemicals can have this potent biological targets. So another area of my research, current ongoing area, is 5G radio frequency radiations. This is a non-chemical stressor, and these are radiations that you see all across, these are radiations that are present all around you, from your Wi-Fi routers to your cell phones. Particularly, my research is focused on cell phone radiation, to the mobile phone antennas, you are basically enshrouded by these kinds of radiations. And particularly 5G has been in the limelight because these frequencies are being deployed. There's like different frequency ranges of 5G. But why this is important to study is that because there haven't been any biological studies with these kinds of radiations, remember, they are non-ionic radiations, which means they don't ionize anything within your body, any molecules within your body, unlike X-rays and other gamma rays, which ionize molecules within your body, despite the radio frequency radiations and non-ionization radiation. But nevertheless, because of the unsurety of their health effects, there have been a lot of controversy regarding their deployment over the last couple of years. And there's been a lot of protest of 5G deployment. And there's been a lot of conspiracy theories, one including very recently that 5G causes coronavirus. Therefore, we need studies to systemically look at whether 5G technology is bad for health. And we are still not there, but using the Zebrafish model, we developed this 5G exposure chamber. So we have this copper box, which is an insulating box with plates containing embryos, and using a transmitter or using an electronic setup, we transmit the 5G radiation signals to these embryos and test whether it leads to any morphological changes, behavioral changes, or gene level changes. And similar to what I explained before, this is the exposure period from 6 to around 48 hours. So these are very short exposures. Then we collect RNA for these gene level studies, and we do some behavioral assessments, and then we grow up these embryos to adult fish to see if there's long-term effects on adult behavior. What we saw with this setup is that the 5G radiations lead to very subtle behavioral effects during embryogenesis with minimal gene, and some of the gene expression studies suggested that there might be metabolic disruption, but this hasn't been tested yet. There were some adult effects, but these were also very, very subtle. So we don't yet have any concrete proof of whether these radiations are harmful or not, but likely they are mostly benign. So just stepping back, going to chemical exposures, what are the uses? Why do we then use zebrafish? So humans are exposed, we all are exposed to thousands of chemicals every day. Within our outdoor environments, we are exposed to air pollutants. Within our indoor environments, we are exposed to dust and other contaminants. If you look at the structure of a sundial, a sand dial here, you see at the top are these human exposures containing thousands of chemicals. Then there are studies which detect and characterize these chemicals using passive sampling or using a blood sampling, followed by mass spec analysis, different mass spec analysis. But now we still have thousands of compounds that we need to test, and it's difficult to test using a rodent model because it would probably take years because they are expensive and they are extensive to test. So we use zebrafish as a screened and molecular biology tool to detect toxic chemicals, kind of in the way that I showed you before, which now that we have identified toxic chemicals, we then do detailed studies on molecular effects and health effects on the identified chemicals. So now we limit our chemical, the number of chemicals, but we are going more wide towards the effects that we are studying, the health effects that we are studying. And then finally, this informs what translates into a community engagement. So these effects need to be reported to the community because ultimately we are measuring human health effects. These will impact remediation strategies, and these will impact other policies. So I hope I could convince you today the application of zebrafish as a model that is used as a model for human health research all around the world. If you have any more questions, I'll be happy to answer this question. And finally, in my last slide, a little bit of nostalgia. These are from my MSc days in environmental sciences. So if you have any questions, I'll be happy to answer them. Thank you.
Thank you, Dr. Dasgupta.
Now the session is open for interactions. Dear students, if you have anything to ask Dr. Dasgupta, please don't hesitate to ask him. Dr. Dasgupta, I would like to ask you something out of my own interest. That has it been studied the genetic effects of heavy metal accumulation in zebrafish in your lab?
Yes, not in my. So our lab, we have done some testing with heavy metals like lead and even some of the radioactive metals. But our lab doesn't focus on metals as of yet. But there's been a lot of studies on not only like morphological or behavioral effects, because some of these metals are really neurotoxic metals, but also like the epigenetic effects. There's been research on whether these metal exposures lead to aging, lead to like accelerate aging, and there's been a lot of other neurological studies. There are an important direction that we are now going into with a lot of research is microbiome studies, so studying how the gut microbial populations affect human health. So there's been a lot of studies using metals and this fish model.
Okay, thank you, Dr. Dasgupta. Any questions from students? Okay, Dr. Dasgupta, hope your insightful talk has enlightened the students regarding the present trends of career options in research and development in environmental science. Thank you very much. Thanks a lot. Hope we will meet again. Okay.
Hi,
Now,
I think there are a few questions
I would
in
like.
the chat box. I think there are a few questions in the chat box. Yeah.
Oh,
Okay.
Dr. Amrit Mitra is asking, please throw some light on the use of zebrafish in drug discovery toxicology. Yeah, similar to my screening, the screening paradigm that I showed, this exact paradigm is used all across the world for testing not only whether a drug is toxic, but whether it is neurotoxic, because a lot of these, particularly these developmental toxicants, are neurotoxic. And the problem with neurotoxicity is oftentimes you don't realize these manifestations do not occur till later in life. So sometimes these lead to behavioral effects or like cognitive deficits, which is not, from a human health perspective, it's not always detected. So it is used, and there's like labs all around the world. In US, we do some of it, but in there's different labs in US and all around the world using these kind of strategies to do drug discovery. There's specific lines of like transgenic, genetically edited lines of human disease. For example, there's zebrafish lines of autism, which can be used to study, whether a drug has an effect on some of these human diseases.
Dr. Dasgupta, there is another question. Which aspects of your master's in environmental science has helped shape or motivate your research?
That's a really great question. So my interest in toxicology basically developed from my master's training, and I always Dr. Krishanu Chakravarti, who was our professor of toxicology within our class, he motivated me to pursue this area of research. And then I did summer training in IICD in Kolkata, working on some of the lab techniques that really motivated me to pursue a career in toxicology. So it's basically an amalgamation of some of the lab techniques I got and some of the classroom teaching that I got during my master's training that led me down this path.
Okay. Thank you, Dr. Dasgupta, for your profound and informative talk. Okay, now I would like to request Dr. Priya Banerjee to take over the session. Dr. Banerjee, over to you. Dr. Banerjee?
Thank you, Dr. Pallavi Mitra, for hosting the previous session, and thank you, Dr. Shubham Dasgupta, for that informative presentation and sharing of all those nostalgic moments with all our audiences this side. It is a delight. It is absolutely a delight to have you as a friend and as a colleague. And I now welcome our next speaker, also my friend and batchmate, Dr. Avisha Chaudhary, who will be presenting another aspect of interdisciplinary research in environmental science. Dr. Chaudhary finished her master's in environmental sciences from University of Calcutta in 2010 and was awarded with her doctorate degree in 2015 from the Department of Chemical Engineering, University of Calcutta. She carried out her postdoc of Biological Sciences, National University of Singapore in 2016. She was also a visiting scientist in Professor Dimopoulos Lab, Immunology Department, Johns Hopkins Bloomberg School of Public Health, United States. She is currently working as a research scientist in Professor Subhas Vasudevan's lab, Department of Emerging Infectious Diseases at Duke NUS Medical School, Singapore. She currently examines the molecular interaction between vector, host, and pathogen for mosquito-borne arboviral diseases like dengue and Zika. So with this, I would like to hand over the session to Dr. Avisha Chaudhary for her presentation. Thank you.
Okay. Is my presentation is visible to everyone?
Yes, absolutely.
Okay. Okay. Hello, everyone. I am Avisha Chaudhary. Thank you, Priya, for that kind introduction. And I would like to thank everyone who have worked to organize today's webinar. Thank you for giving me this opportunity to present my research and ideas with everyone today. Thank you, Shubham, for that wonderful talk. And I hope all of you are doing good, staying safe. Do stay at home and take the vaccine, and ask your parents and others to take the vaccine as soon as possible. So with this, I would start my today's presentation. I have slightly modified the title from what it has been shown in the flyer. So I will be talking mainly about arboviral diseases. By the word arbovirus, these are the viruses which are transmitted by arthropods. So today I will be talking about the mosquito-borne arboviruses. So before going into the main science part of the presentation, similarly like Shubham, I will also show like how I have like ended up today here where I am. So the career path, how it started. So I finished my schooling from St. John's Diocese in Kolkata, and then I did my bachelor's from Ashutosh College with zoology honors. Then I finished my master's in environmental sciences in Baligan Science College under the supervision of the eminent professors, Professor Aniruddha Mukhopadhyay and Professor Punarbasu Chaudhary. And then I finished my PhD with Professor Debarati Mitra from Rajabaja Science College in the Chemical Engineering Department. While I was doing my PhD, I was also serving as a lecturer in the Ashutosh College Environmental Science Department. And then after I finished my PhD, I moved to Singapore. I had to move. It was like a personal thing because I got married and my husband was staying in Singapore. So then you can see all this while already that I had a shift in my research field about which I will be talking to you as well. So once I joined as a postdoc in the NUS, I was also working in collaboration with Professor George Dimopoulos in Johns Hopkins, US. And there I have been fortunate enough to also meet Professor Nobel laureate Peter Agre. So he received his Nobel Prize in Chemistry in 2003 for discovering aquaporins. And then currently I am working in Duke NUS Medical School with Professor Subhash Vasudevan and Dr. Satoru Watanabe. So today's research, what I will be talking about today, has been mostly pursued by me over the last few years. So I have recently joined Duke NUS in February this year. So the work which I will be showing today, they were mostly done with Professor Keeney, Julian Pompon, and George Dimopoulos. So let's start. So let's talk a bit about mosquitoes first. So we are all been annoyed by this creature, right? So these creatures are the smallest organisms, one of the smallest organisms, invertebrate organisms, but they can cause havoc. They are causing havoc. I mean, we are talking about COVID these days, but these are silent creatures which are actually causing millions of deaths and infections annually. And it is not just an emerging thing like COVID has just emerged, but the viruses and the diseases or pathogens this mosquitoes are spreading, they have been there for a while. Like you can date back to 16th century from when you can see that these diseases were there. So these mosquitoes are responsible to cause around 700 million infections as per is recorded by the World Health Organization. They are responsible of causing 700,000 deaths every year around the world. And that is largely because there are around more than 3,500 mosquito species which are present. And what they are doing, they are spreading diseases like dengue, Zika, chikungunya, malaria, Japanese encephalitis, etc. So I will be mainly covering the arboviral diseases, as I mentioned, which are dengue, Zika, and chikungunya in my today's presentation. So let's talk about the two specific primary vectors for these diseases. So the two primary vectors are Aedes aegypti and Aedes albopictus. They primarily cause dengue, Zika, and chikungunya around the world. So this plot here, the graph, the map of the world will show you the prevalence of these mosquitoes around the world. So in the top one, you could see the places where Aedes aegypti is prevalent. These are mainly the tropics and subtropics of the world. And Aedes albopictus is also similarly prevalent in the similar areas like tropics and subtropics. But the difference between these two species in the sense of prevalence is like you could see even in the cooler parts of the world, Aedes albopictus is present, and that is because they are like more cold tolerant species and they are highly invasive in nature. So the diseases which are being reported in from the cooler parts of the world are mainly spread by Aedes albopictus, but Aedes aegypti and albopictus both are like commonly present in the tropics and subtropics. And let's get a few facts about the viruses. So dengue, Zika, and chikungunya are the three viruses that I'm going to talk about today. So dengue and Zika falls under the family Flaviviridae, and they are the type of virus which is termed as flaviviruses. Whereas chikungunya falls under the family Togaviridae, and it is an alpha virus. So as you can see from this facts that now among all these three, dengue is the most reported one, and the most number of infections or deaths are actually caused by dengue around the world. So around more than 400 million infections are reported annually. And as you guys might be aware of, that there are several serotypes of dengue that makes this virus even more complex to study. So dengue is often associated with fetal shock syndrome or hemorrhagic fever. These are like the complex and severe cases of dengue. Although most of the cases, dengue is also like it could also be asymptomatic like for any other viral diseases, but only for the severe dengue cases they could end with shock syndrome or hemorrhagic fever. For Zika, it is like compared to the other two viruses, it has been discovered recently. I think it was discovered in 2007 first, and then in 2013 there was a big outbreak of Zika in French Polynesia, and then we have all we are all aware of that it has been linked to microcephaly in newborn childs, like how we have seen from the data from Brazil. And also Zika is associated to Guillain-Barré syndrome in adults. So this is like a neurological syndrome, have neurological pathology, which happens in adults. For chikungunya, the major outbreak happened in 2004 in Indian Ocean Islands. And since then, around 6 million people have been infected worldwide. And as you can see, for all these viruses, there is no vaccine candidate so far, which is efficient. Now, many of you might be knowing about Dengvaxia. So Dengvaxia is a vaccine introduced by Sanofi Pasteur Lab, but that vaccine has some complexity in it. So although it is being used in some of the countries where you have endemic dengue outbreak, but the major problem with Dengvaxia, which is one of the dengue vaccines present, is that it cannot be used in all age groups. They have a specific limitation of age group, as well as it cannot be used for people who have not been exposed to dengue before. So if they could be only used in people who had an earlier dengue infection. So a person with a primary dengue infection should only get Dengvaxia, not with like a virgin person who was never exposed to dengue in that case. So with this, which is the main challenge here in the field of arboviruses and virology and vaccines as well, that there is a profound research going on, extensive research going on worldwide in different labs where they're trying to come up with vaccines addressing to mitigate these diseases. So the lower two pictures here shows like the genome outlook of these two types of viruses. As I mentioned, dengue and Zika are similar viruses. They are flaviviruses, and they have a one positive single-strand RNA, which can be translated into all 8 proteins. So they have 3 structural genes and 5 non-structural genes, which will be translated into all 8 proteins. And for chikungunya, they have 2 open reading frame. As you could see from the picture, for dengue, there is just 1 open reading frame, which will be translated to one entire polyprotein that will be cleaved into smaller proteins. But for chikungunya, there are 2 open reading frames. So there will be 2 translated into 2 polyproteins, which will be cleaved later stages into all 9 proteins, 5 structural and 4 non-structural. So the structural genes or proteins in the viruses are usually those which helps the virus to get entry inside the cells, helps them to attach to the receptors present on the cells, and then they will get inside the cell through them. And the non-structural proteins are those which helps the virus to replicate and assemble to form the virion particles. So going with this, as I mentioned, there is no vaccine candidate for these diseases. Then how do we mitigate these diseases? For that, the alternative approach could only be controlling the vectors, right? And then when we talk about controlling vectors, there could be several strategies which are being implemented around the world and around different states and places, depending on what kind of amenities or resources are available there. So for vector control method, one of the most common one is using insecticides. But then the usage of insecticide will lead to resistance to the insecticides, and eventually that might not work. So there is another way of controlling the vector, which is being extensively researched around the world, is a biological way to control them. Now, what could be those biological ways? Now, have you ever wondered that these mosquitoes, they are such small creatures, right? And they are also having these viruses, and they are the vectors who are transmitting those viruses to us humans. So then how come like we will develop symptoms, we are even ending with end with like a very poor, unfortunate death we could end up if we have the infections to that some extent of high level. But this mosquitoes being such small creature, invertebrate animals having simpler system, they could actually have persistent infection in their body throughout their life cycle. It doesn't really affect them. I mean, there are reports that they affects them on the way, like they will affect the fecundity. There will be lesser number of egg laid by the female mosquitoes when they're infected. Their life cycle or lifespan is sometimes reduced, but not like a profound effect is there when they have these infections. So what are those factors? There's something present in the mosquitoes, right, which are helping them to have these infections in them lifelong as a persistent infection, but we maybe do not have those factors. So it would be interesting to identify those factors in the mosquito that which are helping them, giving them that antiviral response against these viruses. So if we could identify those factors, and our idea or hypothesis was to identify such factors in mosquitoes and then try to manipulate them in a way and make the mosquito more potent, and so that it could not transmit the viruses to us. So the first step to this research would be to identify those factors in the mosquitoes which are helping them to restrict these infections. So with this, before going, this is just a picture to show how a mosquito insectary exactly looks. This is a picture from Johns Hopkins University. So these are the trays where we actually hatch the eggs. Then when they will metamorphose into from larvae to pupae, then we collect them in the cages and then we rear them. So there is a small video here just to show you. So these are the eggs, then they will hatch into this larvae, and then from the larvae they will metamorphose into pupae stage, and from the pupae they will finally emerge or eclose out to the adult form. So this is how we breed mosquitoes inside the insectary for our research. It is not like we are doing it in the wild all the time. So what we do is we collect some mosquito specimen from the wild, then they are established in proper insectaries, and then we will breed them inside the insectary. So most of the experiments what we do, like infecting them with dengue or any other viruses, are all done in controlled restricted environment inside proper places like insectary, and there are infection rooms like that. So before going into identifying those factors which are present in the mosquitoes helping them to restrict the viruses, we should know something about how these mosquitoes are transmitting the viruses, right? What is the transmission cycle for these arboviruses? So when an infected individual is being bitten by a non-infected mosquito, the non-infected mosquito will suck up the infected blood. Now that infected blood will reach its midgut. What happens inside the midgut is for each virus, when they reach the midgut of the mosquito, they with time they will replicate inside the midgut cells, and finally they will cross the midgut barrier and will reach the hemolymph of the mosquito. So mosquitoes, they have open circulatory system, so they have hemolymph like liquid. How we have closed circulatory system, we have blood, they have hemolymph as their body liquid. So from the midgut, the viruses will breach the midgut barrier and will come out to the hemolymph. Now, the hemolymph will be in the hemolymph, since it's an open circulatory system, it will be spread out entirely into the mosquito body. Finally, these viruses would breach the salivary gland and would end in the salivary gland. Now, when this mosquito now bites another individual, this will be transmitted. So this is like a cycle that when the mosquito bites an individual, sucks in the blood with the virus, from where it reaches the midgut, breaches the midgut barrier, and it should finally come to the salivary gland. This entire pathway is not happening like within hours. It takes time. So that is what is called an extrinsic incubation period for any type of virus. For dengue and Zika, this time for the virus to breach the midgut and come into the salivary gland is around 7 to 14. Usually it's around 10 to 14 days, and for chikungunya virus it's around 2 to 4 days. So after this extrinsic incubation period, when the virus is now finally into the mosquito salivary gland, now when this infected mosquito is biting a healthy individual, it will pass the virus into that healthy individual along with the blood. So when the mosquito is biting us, as when we eat, right? If you take something like a glass or a bowl into your mouth, then you are also transferring some saliva into it. So similarly, when the mosquito is inserting the proboscis in our skin to suck up the blood, in the same time, meanwhile, it will also release some saliva in our skin cells. And through the saliva, they will also transmit the viruses. So what we could see from this transmission cycle, and eventually here the person becomes diseased as well. So what we see eventually here, that mosquito salivary glands are the determining organ, so which is determining the transmission of the virus. So then that is why we focused our research mainly on the mosquito salivary glands, because this is the gland which is actually producing the transmissible viral particles, because the final viruses are replicating and assembling in this organ, which is then coming to the saliva and getting transmitted. So our most of the research was focused on the factors and the molecular crosstalk happening between the virus and the vector genes or factors inside this particular salivary glands of the mosquitoes. So we took several approaches to study this interaction between the vector factors and the viruses. The first one being transcriptomics and proteomics. So for the study, I will just give you an overview of how we designed the experiment. We had control mosquitoes where we fed the mosquitoes just with blood without any virus. We had mosquitoes where we fed them on blood with dengue, with Zika or chikungunya. And after specific extrinsic incubation period for these viruses, we dissected out the glands. So this is how a typical mosquito salivary gland would look. It's like a pair of glands, and they have 3 lobes on each side. And then we collect those glands, and then we will do we are looking for those factors, right, which are helping them to restrict the infection. So we want to look that you will have to see for the changes happening inside the gland. Now, how do we look for changes happening inside a particular gland? We go on to look for the gene expression profiles. So to look for the gene expression profiles, there are 2 strategies. One would be to look at the mRNA level profile, and the other would be to look at the protein level profile. So mRNA are actually is translated from the DNA, the genome, and mRNAs then get translated to proteins. mRNA get transcribed from the genome, and then they get translated into proteins. So we had both the type of samples we wanted, mRNA and protein extracted from the glands. And then after that, we used the 2 cutting edge technologies which are available these days for this omics kind of work. So we used Illumina high-seq sequencing of RNA. So here you could get the RNA sequenced, and from there you could get the number of reads, and then they will eventually tell you which are the genes which were expressed more. So the genes which have more number of mRNAs present are actually transcribed more, right? So those genes are the active ones, we would say. And similarly for proteins, when we extract the proteins, and then we used iTrack labeled proteomics. So this is a kind of method where you use tagged labels to which will tag individual proteins present in your sample. And later, using mass spectrometry, you can actually look at the amount of individual labels tagged to a proteins. That will help, and the software will help you to identify the proteins which were differentially expressed. So using these two technologies, we could identify two things. From RNA sequencing, we can identify genes which were differentially expressed. We call them differentially expressed genes, or DEGs. And from the protein profile, we could see differentially expressed proteins, which are the proteins which were differentially expressed in response to infection. Now, these two result output are always being so we receive this comparing them with the control. So when you have a control, you know which gene is expressing how much in the control mosquito. And when you have an infected one, then you compare between two to see, based from the control, the mosquito with infection, is that gene like expressing more or expressing less. So that's how we define what is differentially expressed gene or differentially expressed proteins. So here is just one of the representative image. I'm not going into the details of all the transcriptome and proteome work we have done. This is just to show you how we usually depict the data. So this is a typical picture of the transcriptomic work what we have done. So on the left, the Venn diagram shows the overlap for the different infections. So where this middle part, which has the overlap for Zika, dengue, and chikungunya, shows these are the genes which were commonly regulated by all the three infections. So there were 6 genes which were commonly upregulated by dengue, Zika, and chikungunya. There were 11 genes which were commonly downregulated, and there were 2 genes, those were differentially regulated, as in that same gene might be upregulated for Zika but might be downregulated for dengue. And on the other parts of this Venn diagram, this is the place where you could see genes which were uniquely regulated by Zika. So they had no effect with dengue or chikungunya infection. This part will tell you the genes which were mostly regulated by chikungunya infection. And then again, on the outer circle, these are the number of genes which were not changed. So as you can see, out of there are around more than 18,000 genes present that we saw from the RNA sequencing. Most of genes were actually not regulated. That means they didn't respond towards infection. So that already streamlined down the work, right? Out of 18,000, you could only see some total of around, taking chickpea, some 1,000 genes which were actually regulated with the infection. And then you have to zoom in further in on those 1,000 genes to check that which are the exact factors affecting or responding to this particular infection. And on the right side, we have categorized these genes based on their functional classes. So some genes, as you can see, will be apoptotic genes. Some genes were genes responsible for the mosquito blood feeding. There were genes responsible for mosquito immunity. So this plot here shows the number of genes which were upregulated with the infection, and these bottom part shows the number of genes which were downregulated with infection. Now, we were more interested, this is a huge data set when you work with RNA sequencing or when you work with proteomics, but you have to really streamline down your work. So what we were more interested is to look into the immune genes for the mosquito, because as I mentioned before, mosquitoes can restrict the infections, and it is mostly the immune system for a particular organism which helps the organism to restrict any invading pathogen. So we were more interested to look into the immunity genes first. So what we know so far about mosquito immunity is highly abridged from what we know in Drosophila. So Drosophila is a model organism, and most of the work is usually done in Drosophila first, and then they were subsequently being tried in other flies or insects. So what we know from Drosophila is the 4 main insect immune pathways present are toll, IMD, JAK-STAT, and RNAi. I won't be going into the details of this pathway. I just wanted to mention that the pathways that we know are present in the mosquitoes or present in Drosophila, and we know they might be also present in mosquito. So these are the 4 main pathways acting in them. Insects, they don't have adaptive immunity, they just have innate immunity. Now, our next idea was to check that, as I told you, we had identified immune genes which were differentially regulated. So now we wanted to functionally classify those genes, that what happens if we remove those genes from the mosquito or downregulate their expression. So our first method was to do a gene knockdown. When we say gene knockdown, we are trying to reduce the expression of a particular gene and then infect the mosquitoes and then see what happens. So if that gene has some particular function in responding against the infection, then when you remove the gene or remove the protein from the organism, then that protein cannot function properly. So then you could see that what happens, you could see a phenotype in the mosquito. So to do a gene knockdown, there is a method where we use double-stranded RNA. We inject the double-stranded RNA inside the mosquito under microscope. That it's a painstaking work, but we have to do it. And then after that, after a specific time, the double-stranded RNA, actually double-stranded based RNA silencing is a method. I mean, you guys can look into the internet. It'll take a longer time if I try to explain it here. Is it basically works on RNAi, so which is RNA interference pathway. So you put the double-stranded RNA, and the RNA will complementary bind to the RNA present for the mRNA of the gene present in the mosquito, and then after the complemented binding, there is an enzyme Argonaut which will cleave the mRNA, and that's how you make the mRNA incompatible, and there will be no more translation. You make it inactive, basically. So that is how you knock down the gene in a particular mosquito. And this is the data what we found is we tried to knock down several genes in the mosquito salivary glands, and comparing it with the control, I would just explain one of them. As you could see, when you comparing it with the control, when you knock down the gene, if you see that the viral numbers are going up, that means when the gene is not present, the viruses are going up. That means when the gene is present, it is helping to restrict the infection. So that's why when you remove the hindrance from the pathway, if you remove the gene which is actually helping the mosquito to restrict, when you knock that gene down, then you could see viruses replicates more. So this is how these data are being read, that compared to the control, you could see for this gene, which was kayak, when we knocked down this gene, the gRNA copy numbers went up. Similarly, for this, we did for dengue, we did the same for Zika and chikungunya, and we found several proteins which actually showed more viral copy numbers when they were knocked down. And we could also find another type of protein or genes which is proviral. So which ones are proviral? So when we knock them down compared to the control, you see a suppression of gRNA. So that means when these genes or proteins are present, they are actually helping the viral replication. So that's how we identify antivirals and proviral factors in the mosquitoes. We also found out by microscopy that there are a few genes present in the mosquitoes which helps to reduce the virus by causing apoptosis. Now, drunk has been reported to cause apoptosis in vertebrates as well, but it was never being validated in mosquitoes if it is the same a gene which is causing apoptosis. In this particular image I'm showing here, this is a fluorescence microscopy of salivary gland of mosquito, and you could see the first three picture shows the control condition, and this is a down regulation or knockdown of the gene called drunk. So when we knocked down the drunk gene, here we are staining for apoptotic factors, or sorry, apoptotic bodies. So apoptotic bodies are those. So apoptosis, as you might be knowing, are programmed cell death. So when a cell is infected, it is like a suicide of a cell. Sometimes when they're infected, they will kill themselves. So you could see this particular gene drunk, when we knocked down that gene, we couldn't see apoptosis. But when this was present in the control experiments, there were a lot of apoptosis happening in infected salivary glands. So this is how we also revealed that drunk is also responsible for causing apoptosis, which in mosquitoes actually helps in reducing the infection in them. So another strategy which we tried, which I worked with in Johns Hopkins in collaboration, is based on the CRISPR-Cas9 technology. Now we know that how CRISPR-Cas9 has the two famous scientists, Emmanuel Charpentier and Jennifer Doudna. They received the Nobel Prize back just last year in 2020, but for the breakthrough of CRISPR-Cas9 technology. But then CRISPR-Cas9 was identified as early as in 1987. And then with time, scientists started to follow the pathway, how it works. And eventually by 2015, they could finally fully enlighten us with exactly how CRISPR-Cas9 works. Now, CRISPR-Cas9 is a system which is present in bacteria, which is an immune system for them. So it acts as a memory system for them. So whichever invading pathogen they faces, they will integrate a small part of that invading pathogen inside their own genome. And next time when the virus invades that same bacteria again, then from this immune pathway, they will identify the particular area because they already have a part of that virus. So they will translate again they will transcribe the RNA from that particular part on their genome, and that RNA will come and bind to the virus now and will be cleaved by the Cas9. I mean, CRISPR-Cas9 also is a very known and highly explained technology. Whoever wants to know in detail about it can always study. There's a lot of information in literature. So just a basic outlook of how it is happening. So basically for CRISPR-Cas9, so it was identified as an immune system in bacteria, right? But these people, they have implemented it into gene editing tool. So how it is editing the gene. So for editing a gene with this technology, you have to design a guide RNA, and you have to have, there are basically two components. You have to have a guide RNA, and you have to have the Cas9 protein, which will be acting like the scissors. So when you design the guide RNA along with the Cas9, when you introduce it into the cell, the target DNA you want to cleave, the guide RNA will go and bind complementary to the particular part of that gene, and the Cas9 will then cleave the genome. And that's how you can make that particular gene inactive, because now the gene is cut, so it will not form the true protein, which canonical true protein which it was supposed to be translating to. So, and this another thing what you can do is either you can knock out a gene by cutting it from the middle, or you can introduce another gene inside. So once you cut it and then you introduce another designed RNA transcript, which will come and will be binding inside that particular region by homology directed repair. So now the gene you wanted to introduce, it could be from another organism, it could be from other part of the same genome of the organism, but you just want to reintroduce it into the system. So all this kind of playing and manipulating the genome could be done using CRISPR-Cas9. So we had recently this authors in Kistler, they published this work. They had tried it in mosquito and they could do it successfully. So they had so they will design guide RNA, and with the help of Cas9, they will it can be cleaved at a particular part, and whichever gene you want to modify, you can do it. There are a few outcomes for the CRISPR-Cas9. There could be, as I mentioned, there could be deletion. You can just delete a part of the genome gene completely. You can introduce something which is called homology directed repair. You can introduce something of your choice in that particular region, or you can also delete a bigger chunk, like from some genes are really big. So usually when we do CRISPR-Cas9, we put one small guide RNA of around 20 nucleotides. But if you want to remove a bigger chunk or bigger segment from the genome, then you have to introduce multiple guide RNAs, which will cleave out two distinct positions, and that entire chunk in the between will be lost. So several kinds of things could be done with CRISPR-Cas9. So for CRISPR-Cas9, the main challenging part for doing it in mosquito is that you have to inject them inside the mosquito eggs. Now, if you remember in the one of those video which I showed you, the mosquito eggs in naked eye is just like a black dot. But when under microscope, they looks like this, small like rice grains. So with time, after from the mosquito lays the eggs, with time they will slowly melanize to become dark melanized eggs. So there is a particular timing within this 30 to 45 minutes while the egg is turning into a melanized one, that you have to inject the mosquito embryo under microscope using microinjector. So which is usually around somewhere here, where it should be like mid-grade to a grade time. So if you are too early, then also you cannot have a proper CRISPR-Cas technology happening, or if you do it too late, then also the gene editing won't happen. So this particular, capturing it in this particular time and injecting the mosquitoes is really challenging. But then people have done it, we have done it, so it is an achievable job. So we have done a proof of principle work where we tried to knock out one gene, which I worked in George's lab in the US. So initially, after injection, you will first get a heterozygous line, and then after getting the heterozygous line, you have to mate them to get the homozygous line. This is how it will follow the Mendelian inheritance law, right? First, it will be heterozygous, then when you cross heterozygous, then you will get the homozygous clones. And this is proof that how we have successfully knocked out a part of the gene. So we had the gene we worked with is called IKK2 or IKK gamma, is a regulator of one of the immune pathways in mosquito. So this is a picture of an agarose gel. So on an agarose gel, when you run DNA, they will show you the bands or the sizes of the DNA. So here what I am trying to show is for wild type mosquitoes, the gene size is bigger, and for knockout mosquito, this gene size has reduced because it has traveled further down. So these are smaller gene. So that means this difference in the size is that particular part which has been deleted from the genome. And that particular size, when we sequenced, we found out that the deletion part was around 22 nucleotides, and we confirmed that this gene was successfully knocked out. A part of the gene was actually removed from the entire gene, and which makes the gene non-functional. And then we also did a small experiment preliminary to see if what happens when you knocked out the gene. So we had data before where we had knocked down the gene, where you just reduce the gene expression. But here we have completely knocked out the gene. So now there is no protein for this gene at all. And then we could see that for this particular gene, IKK2, we could see that it actually is susceptible. It makes the mosquitoes more susceptible to dengue. So when the gene is not totally knocked out and you infect them with the dengue, we could see that actually the GRN copy numbers were much higher compared to control, which tells that IKK2 is one of the main antiviral factors. So when you remove it, the mosquitoes are becoming more susceptible and dangerous to infect and transmit the virus. So this is how these are the different type of technologies. I'm trying to, like, in today's talk, I'm definitely trying to give ideas about the technologies which are available and you can use. It is not just in vector biology, you can use it. If you search this all in other biological fields, you could use all these technologies for gene knockdown or gene knockout. And then currently I'm also working in my new position with Professor Subhash Vasudevan. I'm also working to identify and compare the different strains of dengue in Singapore. So dengue in Singapore is quite prevalent. There has been major breakouts in dengue in Singapore over the timeline. There's a major breakout was at 2013. Then the most biggest outbreak was actually last year. So now we are trying to compare the different strains over these different outbreaks and see what caused those outbreaks. I mean, what were the factors, or how were those strains of viruses more infectious compared to when we didn't have an outbreak. So this is the kind of project I am currently into. And also I'm working on dengue and Zika in mice. So as we know, I said that we know that it causes microencephaly in human newborns, but then we are trying to see what happens in mice. We will be working on the mice placenta, the decidua and the fetal placenta, and see if dengue and Zika, what are the factors present inside the placenta that helps in the transmission of this vertical transmission of this dengue or Zika from the mother's womb into the fetus of the child. So these are the very new projects. I'm just giving an overview that what are the ideas of projects we might start looking into, which are not known yet, that how were they caused or why are they caused. There is also one very interesting project which is currently being implemented in Singapore and Australia and a few other countries in the world, which is Wolbachia infected mosquito. So Wolbachia is one of the bacteria. When it is present in the mosquito, it makes the mosquito less susceptible to the virus, because Wolbachia being a bacteria, it competes with the virus, and either one of them can infect the mosquito properly. So it has been seen that when Wolbachia is present in the mosquito, then those mosquitoes cannot transmit the dengue or Zika or other arboviruses. So currently there's a program where they have introduced Wolbachia inside the mosquito, and then they let those mosquitoes mate with the wild type females. So basically they inject the Wolbachia in male mosquitoes, and when those Wolbachia infected male mosquitoes mate with wild type females, they cannot produce viable eggs. So those eggs cannot hatch. So both ways they can actually restrict the spread of the disease. So these male mosquitoes, when mate with the females from the wild type, then the eggs, whatever they hatch, they don't hatch. They lay, what they don't hatch, so they are sterile. So the mosquito are basically sterile, the males. So this is a very nice strategy to introduce mosquitoes in the wild. They are not genetically modified in a way, because you are just introducing the viruses, the pathogens inside the mosquito, and making it more incompetent for virus transmission. So this is one technology which is being used in Singapore and other places to curb or mitigate the spread of these viral diseases. And talking about what we could do from this research, I tried to put it in this way. So in our project, so when we talk about arboviral diseases, it's basically if you see a three-way interaction. So there are three main components: the vector, which is transmitting, the pathogen, which are being transmitted, and they are being transmitted to humans, or here we call host, because we study most of the human studies are basically done on the mouse. So this three-way interaction has a lot of possibilities to study, and we know very little about it. So this mosquito saliva is actually the main center point for all these studies. Like for currently in my own research, I'm mainly focusing on vector and pathogen interaction, where I'm trying to understand the barrier factors which are restricting the infection. And then we could make the mosquitoes, like we can make transgenic lines to make the mosquitoes refractories. I have my colleagues and my other friends who are studying the interaction between vectors and the host, where they can identify factors. So if you don't talk about viruses at all, mosquito saliva itself has some particular interesting factors in it, like molecules which are vasodilatory and anticoagulant. So as you can know, when mosquitoes sucks our blood, they get a steady flow of blood without any hindrance. So there are particular factors in the saliva which always tries to dilute the blood as they suck it. So those factors are the dialants, they are the vasodilators, they are the anticoagulants, which helps the mosquitoes to suck the blood. So if we could identify and get those proteins out of their saliva and produce them, then those could be novel therapeutics, could be used in patients where they face blood clots, in cardiovascular disease patients. We could use those drugs. Those could be the novel therapeutics coming out from mosquito. There are many studies done from ticks, where they have identified such factors in tick saliva and have been modified and implemented to form novel therapeutics. And on the third angle, when you talk about the pathogen and mouse directly, then this is the direct vaccine candidate research, where you are trying to identify the interaction, that how the virus is interacting inside the host cell. And then you can identify factors, because there are particular viral proteins which are responsible for causing infection in the mouse or in the humans, right? So to identify those epitopes or those exact factors present on the viruses would lead you to the vaccine research, because in vaccine research you need those factors to be identified on the viral surfaces against whom you are going to make antibodies, or you will immune your system. So in vaccine, maybe you guys already know that we attenuate viruses and we put it just with those proteins. We don't put infectious virus in vaccines. So many of the vaccines are just inactivated viruses. So to make those inactivated viruses, you need to identify the proteins on the viral surfaces which are responsible for the cell entry. So all these are the different aspects of this saliva-centric research, mosquito-borne research. So there are several angles. Several angles are not known yet. A lot of research is possible in this area, and actually this area is quite underwhelmed and unappreciated in a way, because the main limitation is to have a mosquito insectary, because that is like a huge arrangement in itself to have a big insectary and the proper environment to do all this work. So there are like not many insectaries around the world and not many research labs doing work on this particular host and vector interaction. So with this, I would like to thank all my PIs. I mean, I have introduced them already before. I have been fortunate enough to work with all these eminent scientists around the world, and I've learned a lot from them, still learning. And I would like to thank National University of Singapore, Ministry of Education in Singapore, and UCANUS for funding all this research work for all these years. And with this, I would like to thank all of you for your kind attention, and I would be happy if you have any questions for me.
Thank you, Dr. Chaudhary, for the wonderful session, and we are now open for the question answer. You may have seen a suggestion from Dr. Amrit Mitra, who suggests you to use an alternative of CRISPR-Cas9 to get a better perspective, that is Cas-Clover. So can you tell us anything about that, whether you have used it or not?
Yeah, no, sorry, we haven't tried that yet. We have just, as I mentioned, it was a proof of principle, because we have, I just introduced this technology in our lab here. So I went to US to basically learn the technology and then come and introduce in Singapore. So currently we are just working with CRISPR-Cas9. Haven't tried that, but that would be interesting to try it in future.
Okay, so there's a question from Orindom who asks, how do Hematophagus arthropods spread arboviruses, especially dengue, through Trans-Ovarian and Venereal transmission?
Okay, I would like to have a detailed explanation of Trans-Ovarian and Venereal transmission and what those exact words mean. I am not really familiar with those words. If the person can explain a bit more.
Venereal, I guess, means is a sexually transmitted disease, if I'm not mistaken, and Trans-Ovarian means it's supposed through the fertilization process. So I'm not very familiar with the terms, but is there any way this thing could be transmitted through any sort of
I mean, Zika definitely can. There is a report for dengue as well. If you're talking about sexual transmission, Zika, it could be sexually transmitted. But dengue, there is a report. There are a few reports, but it has not been like properly validated. I mean, there are questions about those reports because, like for you trust information when it's happening worldwide with among many people, right? But the information about dengue was just from one or two places, so it wasn't really clear that it was because of sexual contact or it was just through blood transmission. But yeah, Zika can definitely be transmitted sexually. Yeah.
Okay. I would like to ask you a question on behalf of all the students. That is, because there's this pandemic on, and students are mostly restricted to their houses. Their academic activities have been restricted to online activities. So, what other skills can they gain to, train themselves for international research or pursuing research in India as well? So, what sort of other skills do you think they should gain other than their academic knowledge?
I mean, one thing at the top of my head is coming is definitely they should start learning bioinformatics because that's like the thing in the future. It's already a thing now, and we are like learning it. But I mean, people have started to look at. They want researchers who already know bioinformatics to the core. So when we talk about bioinformatics, it mainly covers those analyzing of RNA sequencing data or proteomics data. There are single cell sequencing happening these days. So these are the real in thing. The fashion in science is now bioinformatics. So they should really start looking and study bioinformatics as soon as they can, because people who are trying to stay in biology definitely would require bioinformatics at any point of their research. There's no way out from it.
Thank you,
Yeah.
Dr. Das. Okay, would you like to say anything?
Yeah, I have a quick comment and a question. So I totally agree with Dr. Chaudhary's comment on bioinformatics. It's the skill set that everyone needs right now, like computer coding and coding in R, coding on Python, and learning all the bioinformatics. My question regarding your presentation, first of all, really awesome presentation. This was, having suffered from dengue myself, Dr. Banerjee knows that. It was a really great presentation. So you said that the virus is transmitted through the gut. Do you know or think if there is an impact of gut microbiomes on the virus? Because these are all like microbes, right? So gut microbiomes, or like if altering the microbiota would be an effective strategy. And this is coming from a point of view of not knowing about microbiome too much.
Yeah, thank you. That's an interesting question. Definitely, the microbiome of mosquitoes, it affects, but it's very difficult to control that. As I said, the Wolbachia thingy, which has been implemented, is also based on the Wolbachia is also present in the microbiome. So we put that, and that will compete and reduce the infection in the mosquito. So any kind of microbiome, there are studies where they have tried and identified different microbes which are sometimes acting as a agonist for the viral replication, or sometimes an antagonist. So, but yeah, there is no clear idea exactly that, because if you completely remove the microbiome from mosquito, the mosquito dies, because as the microbes are important, we have microbiome and it helps us. Similarly, it helps them as well for survival. So it's very difficult to control and experiment exactly which microbiome you can remove and check. I mean, yeah, there are studies going on, but not any concrete idea we have at the moment.
Thank you so much.
Thank you, Dr. Dasgupta and Dr. Chaudhary, for those brainstorming sessions, and thank you for encouraging and motivating our students who are presently carrying out their curriculum from their home. If nobody else has any questions, I'd like to hand over to Dr. Shantanu Chakraborty, coordinator of Environmental Science, Rani Rashmoni Green University, for the formal vote of thanks. So, over to you.
Thank you, Bia. Thank you all for this wonderful webinar from which we could learn a lot about emerging trends and future prospects of environmental science research. Now it's the time to officially accord our vote of thanks. At first, I am thankful to Professor Ashutosh Ghosh, the Honorable Vice Chancellor, Rani Rashmoni University, for his support. I would also like to convey our sincere thanks to Dr. Aniruddha Mukhopadhyay, Professor and Head, Department of Environmental Science, Calcutta University, for his presence and support in all endeavors. My sincere gratitude to Dr. Avisha Chaudhary of National University of Singapore Medical School and Dr. Shubham Dasgupta, Oregon State University, USA, for their really illuminating lectures in this webinar. A lot of thanks goes to Onumita Sarkar, Assistant Professor of Botany, and Shoikat Bandyopadhyay, Assistant Professor of Philosophy, Government General Degree College, Singur, for their flawless management of this webinar. Thanks to Dr. Amrit Mitro, Assistant Professor of Chemistry, Government General Degree College, Singur, for his support as always. I have no words to thank Dr. Priya Banerjee and Dr. Pallavi Mitro of Rabindra Bharati University, who had the keys in organizing this webinar. Lastly, my heartfelt thanks to the participants of this webinar and the students of Rani Rashmoni Green University, for whom the program was organized. Hope all of you have submitted your feedbacks in the prescribed format given in the chat box. We promise we will send your certificates within a week. Finally, stay safe, stay at home, and tie hands together from distance, of course, to stop the menace of COVID-19. Thank you very much. With this, I think we should conclude our session for today. Thank you very much, Dr. Avisha Chaudhary and Dr. Shubham Dasgupta, once again for accepting our invitation and delivering such a lucid and very illuminating lectures to our students. Thank you very much.
Thank you so much for hosting us.
Thank you. So, I suppose we can leave now, sir?
Yes. I once again request all of the participants to fill in the forms, the feedback forms, so that we can prepare your certificates and deliver you in time. Thank you very much.